reasons how it can occur. A crucial step in long-term cellular responses is a transient jump in intracellular redox state into more oxidase direction. Yeah, as I told, that it can work in different types of cells, proliferation model, excitable cells, in lymphocytes priming, for example. And I would like to finish just to tell that some years ago my last book appeared, and if somebody is interested in this science, you can find most of those things in this book as well. Thank you for your attention.
Thank you very much for your very informative and stimulatory lectures, and I believe the Professor Karu's lecture re-strengthened the basic understanding or basic theory of the photoactivation of the laser treatment. So time is very limited, but I would like to ask you one or two comments. First, yeah.
Quick question, Dr. Karu, would you extend your conclusions to light with wavelengths and the 900, 910, 905, 940, so on?
At least what I know, that it works till 980. And now it is considered more or less that after 950 the effects are weak. And the reason why one can work with many wavelengths, not only these 4 ones which are in peaks, it means the reason is that the bands are very, not narrow, they are very wide, and there are almost overlapping in this. And even when you irradiate, maximum is somewhere here, you irradiate into this part, for example, of this absorption band, you get some biological response, not so strong like when you irradiate the maximum of absorption band, but you get it.
Professor Hatsumi, and then Professor Kaplan.
Yeah,
okay, Professor Kaplan, please. Yeah.
Tiina, I have a problem with mitochondrial myopathy, where, of course, as you know, the cells fail to produce ATP. What have you done about it?
Yeah, they produce it, and under irradiation they produce it much better, much more. This was the first thing, really, and we did that, and many other people before us and after us did that, measuring ATP concentration, ATP amount per cell after irradiation, and there was a nice increasing in this. Yeah. It is the most important thing, not only the regulation of signalization, it is important for understanding mechanism, but ATP, of course, it is increasing. Yeah.
Do you know of anybody who has clinical experience with cases like this?
Clinically measuring ATP?
With mitochondrial myopathy?
I don't know. But I think there are people are connecting clinical work with taking examples from the tissue and so.
Thank you.
Thank you, Isaac.
Final question, yeah, okay. Professor Katami.
My understanding is correct, and your hypothesis just booster effect of the low power laser therapy effect, booster effect to transduce of the effect level, summary effect level in the mitochondria, and this introduce of the biochemical reaction in this respiratory chain. So this is your hypothesis. So if this is correct, infrared spectrum is more effective than the other spectrum. Infrared is more effective.
You mean that mitochondrial wave is more effective?
Yeah, heating effect.
heating, yeah.
Inside of the mitochondria.
I didn't speak
Yeah.
about this. It all depends which intensities and doses you use. And of course, every work, when starting with some new object, some new cell culture or what else, you have to check the dose dependencies, intensity dependencies. There are doses like this, intensity dependencies is like this in those cases. And of course, those results, they are performed with optimal parameters of light for this or that spatial model. But yeah, this is the first step, of course, because in some cases you can get some nice inhibition as well when your intensity is very high, for example. It means once more that one must be careful taking into account wavelengths, dose, intensity. Those are three most important parameters before you start experiment.
Okay, thank you.
Time is coming. I'd like to close this lectures. Thank you very much,
Thank you.
Professor Karu. Thank you.